Good morning, everyone. Hopefully everybody's enjoying this nice, sunny, dry, allergy-inducing weather. I know my allergies just went off scale the last day or so, but in terms of fighting it, think glucocorticoids and you'll be in good shape. Kills mast cells and things like that help you, allows you not to respond as well to the allergies. It was a terrible sports day yesterday in the Bay Area, so I won't talk about it. And also, I know most of you guys have a lab practical coming up next week. Is that true? All right, because I know next week is always the hardest week to teach because you guys are so stressed and everything, understandably. But just remember, let the adrenaline work for you. Let the epinephrine work for you, and you'll feel a little better about it. Okay, so we will continue today, briefly finish off inductive signals, and then get into digestion. So by the end of today, you'll know a lot about going from food to waste. Okay, as a brief review. Oh yeah, one other thing. I'll have a question and answer review-like session on Wednesday, May 2nd, 10 to 11:30, and I think it's 2050 VLSB. You don't have to memorize that at the moment. You have a lot on your platter. I will send out a B-Space announcement along with some practice questions. Again, as I mentioned on Wednesday, for my questions, for my part of the exam, Mike, how many points do I have total? 500 or something? Or? Okay, according to Mike, I have 178 points. He's the point guru. Under none of my questions, the answers will ever be all the above, none of the above, A plus B, every other Tuesday type things. Educationally, it is not a good idea. Those kind of questions aren't as useful as actually knowing the answer. So there'll be one unambiguous answer for every question, at least I hope. That's the goal. But it's been pretty good in the past. So that's the thing. But I'll send you guys practice questions by B-Space next week, because I know you're all dealing with the lab practical stuff. And you guys know this stuff better than most people in the world right now. So keep rolling with it. So you get all that information next week. All right. And also office hours today, 9:15 to 10. Review. Frog versus mammalian cleavage reactions. What you should do is know, in the comparison of these guys, cleavage planes, gray crescents, blastula, blastocyst, morula, blastocele, compaction, polarity, partitioning of the cytoplasm. You guys I should be able to say to you compaction. You should be able to immediately say what? Mammalian right at the 8-cell stage, right? Doesn't happen in frogs. And it's we're contrasting and comparing frogs versus humans because frogs are a good experimental system historically. And so that's why it's not as arbitrary of a comparison as you might think. But in the lab, I know you guys know this stuff much more detailed than I've presented here. But for my exam, you've got to know those kinds of issues. All right? Gastrulation, forming of a 3-layered embryo. Those 3 layers are endoderm, ectoderm, mesoderm. And you don't need to memorize every tissue that ever comes from the mesoderm, ectoderm, endoderm. One way to think about it, you don't have to know this, endoderm, inside intestines. And we'll talk a lot about that today. Ectoderm, outside skin, mesoderm, in the middle muscle. Okay? So if you have a sense of those organ systems, you have a sense of the significance of endoderm, ectoderm, mesoderm. Frog form, all forms that are blastopore. Mammalian and surprisingly birds use a primitive streak. The only difference is one is a hole or a pore, the other is like a big seam that the cells start moving. And that's really the key difference there. We didn't get into too much detail. Those of you taking developmental biology classes in the future will get into this much more detail. Okay? Organogenesis, morphogenesis. Organogenesis, formation of rudimentary organs, kind of from the name. Morphogenesis, the shape of the organism. The driving forces are changes. It's a mechanical kind of thing besides proliferation. And the driving forces are cell shape changes, cell-cell interactions, and cell movement. And I just gave you a couple interesting examples. Cell wedging, where the cells widen in the bottom. Because of that, you can form the neural tube and neural crest formation. So it's just the shape of the cells changing in unison changes the shape, and therefore the beginnings of organogenesis. Convergent extension allows you to elongate tissue. So you can get a smaller fat tissue to a longer, skinnier tissue through convergent extension, which is just cell movement and cell-cell contact. And then directional cues controls the cell movement. The movement of these cells is not random, and you have directional cues as to where to go, when to go. And a lot of these directional cues tend to be extracellular matrix components. Okay? And we use the classroom here for examples of understanding some of this. Questions on any of that? Okay? Yes?
Are you saying that blastocyst applies to frogs and not mammals?
No.
Then which side is frog?
There is no side. You need to know blastocyst, attachment. A cyst is humans. Okay, you guys should know that. Blastula, there's no cyst. There's no attachment. It's a frog because it's out there. It's happening in the water, in the pond. You got to know differences in those, 9 concepts, comparing them to frog versus human. So, no, they're not in any order. Correct. They're not even in alphabetical order either. I don't think alphabetically, so that's why. All right? If I said to you, blastocele, frog or mammals? Both. That's the answer. There we go. Yes? Can have twins? Well, it depends on your definition of twins. I mean, one can argue when you have a frog, you've got a million fraternal twins out there, right? Because it's all separate fertilizations and eggs. But in the human, mammals, obviously, because the way it's rare, because a lot of animals, I don't know all the exact physiology, but cats and dogs can have multiple births, for example. And humans generally ovulate one. So for us, it's really a big deal when you have more than one individual being born. That's all. But animals, it's multi for a variety of reasons. I think one of the primates, there's a lot of twinning in primates, and also certain kinds of canine species that are out there can have two. Bear cubs, right? Bear cubs is another one. So for some animals, it's more prevalent than others. But the issues are the same. Usually out there, you can have identical twins in theory. Okay? And hyenas, that's the one I was thinking of, where you have twins, but every so often you get 3 come out. And there, the females beat up on the other ones. So they're the much more aggressive of the two of the genders there. Okay? It's kind of interesting if you guys. It's been studied here at Berkeley for a long time. All right? But you only need to know twinning vis-a-vis humans. And you should know the difference between fraternal, identical, and mirror image identical. Okay? All right. Inductive signals. There are positive and negative. Regulation can be up or down. For a positive signal, you have a certain phenotype without a signal. And then when you add a positive signal, you now have a phenotype. In this drawing, it's a square. I'm not trying to say anything about the shape of the cell. It's just a phenotype change. It's an illustration of a phenotype change. Okay, there's a difference. In a negative signal, you block a positive signal. So here, you have a positive signal, keeps them round, but the negative signal blocks it. So the phenotype without a signal, in this case, is a square. Where for the positive, for the other analogy, the phenotype with a signal is a square. So the end result is potentially the same, but it'll differ whether you have a signal or not. And therefore, there's a difference if it's a positive or negative signal. Okay, does everybody see that? Because I'll give you a specific example of each of these in a second. Any questions? That's where we left off. Okay, so negative signals. If I can find the eraser, I'll be in good shape here. All right. So the way to think about this is there's the most famous negative signal comes from an area in the developing embryo called the Speeman organizer. And this dictates dorsal. Okay, so dorsal is back, your back, versus ventral, which is your belly. And it comes from an area, remember I said the gray crescent specifies underneath it dorsal polarity? Later on in development, that area under the great crescent, or what was the great crescent, develops into the Speeman organizer. And it's all based on concentrations. So what happens is you have a ventral signal, which is belly, and this positive signal forms ventral. Okay? And you have a concentration. I'll just cut this off here a little bit. I'll do this down here in a second. In contrast, you have a dorsal signal, which is a negative signal. So where the negative signal wins out over the positive signal, you block it, just like I have in the diagram there. And when you have no signal, you have dorsal. When you have a positive signal, you have ventral. So what the dorsal polarity signal is doing is it's blocking out what the ventral does. So one way to think of it, we'll get to the specifics here in a second. So this is a concentration curve. Here's the ventral signal. As you start from this side of this developing embryo, this is higher concentration and lower concentration of this positive signal. In contrast, you've got this dorsal negative signal that comes from the Speeman organizer. And you get higher concentrations. So right about here, approximately, dorsal is greater than ventral, right? The negative signal is higher concentration than the positive, so therefore you block it out. And when you block it out, you have what? No signal, right? Because dorsal is inhibiting. So dorsal is inhibiting the ventral signal, which gives you dorsal, which is, in this case, no signal, right? Because you're preventing the ventral from working. So the default pathway is dorsal. Over here, who's higher, ventral or dorsal? Ventral, right. So here, ventral is greater than dorsal. So you can have all the negative signal you want, but the ventral positive signal wins out, and it becomes a belly. Does everybody see that? Any questions? So it's concentration gradients coming from either end. And whoever wins out at a point in time, or a point in spatial time, I should say, it dictates whether you have the ventral signal or you block the ventral signal. And when you block the ventral signal, you look dorsal. So it's just exactly as I have drawn here with this negative signal. So in this case, this is ventral, and then this is the dorsal signal. Okay? Question? Yes?
Can't we think of like the dorsal signal being positive and ventral signal blocking it?
In this case, no. Because if you had the dorsal signal there with nothing else, there's nothing. It's a default pathway. It's like having no signal. Okay. What the dorsal, in this example, and I'll give you it might become slightly clear when I give you the actual molecules involved. They know what these are. What the dorsal signal does, its role is to block the ventral signal. Okay, that's its role. That's the same thing as having no signal whatsoever. In both cases, you end up having a back. So for example, if you took a developing embryo, okay, and you have this ventral signal coming in, the dorsal negative signal coming in, and you added another dorsal signal here. You add another inhibitor here. Very high concentrations. What would you predict about the polarity of this developing embryo if you added a second dorsal signal, dorsal negative signal to this developing embryo? What would you predict? Well, more dorsal, right? And when you do that, normally, here's the normal embryo, dorsal ventral. What you get is, if I can draw this, two dorsals with a little tiny bit of ventral in there. Okay? So it shows you can just block it out. It's clearly acting on it. All right? Questions? So what these are, they actually know what these signals are. And I'll put this up here. The ventral signal, whew, that was a good one. Haven't broken these blackboards yet, but time can still happen. All right, so the ventral signal is bone morphogenic protein 4. BMP4. And don't worry about the name. It got discovered originally as a helping out, or its family of proteins, laws for bone development, and the name stuck. But this is the ventral signal. So what happens is BMP4 binds to a receptor, depending on the concentration gradient, so it works in here, right? And when it's in high enough concentration, the response is ventral structure, or bellies. Okay, everybody see that? I'll do this over here. The dorsal signal, dorsal negative signal, is noggin. This gene was actually discovered here at Berkeley, Richard Harlan's laboratory. And what noggin does is it directly binds to BMP4 and prevents it from interacting with its receptor. So you have BMP4 here, and noggin comes in and actually directly interacts with it and creates a protein-protein interaction. As a result of this, BMP4 cannot find its receptor. And when you don't activate the ventral receptor, you become dorsal. So in essence, the dorsal signal here, this negative inductive signal, is an antagonist of BMP4. But instead of interacting with the receptor, sort of in a classical antagonist manner, it binds to BMP4 directly and prevents its interaction with the receptor. Everybody see that? Noggin binds directly to BMP4, prevents BMP4 from interacting with its receptor. Yes, as I just said, it's a form of an antagonistic kind of action. But instead of binding the receptor directly, it interacts with the ligand or the binding proteins. Everybody see that? And that's how it works. So you can take noggin in there and inject it into these embryos and get the same thing. And that's how it works. It's an antagonist. Now, positive inductive signal, a good example of a positive inductive signal is ZPA. I mean, another example is this ventral. BMP4 is a good example. But I think this is an interesting one. What? Zone of polarizing activity. And this dictates, that's good. This dictates ZPA in the mouse limb bud, dictates posterior digit formation. So what are posterior digits? Those of you guys have this in your lab thing, polarity and stuff. How do you know which digits are posterior or anterior? Well, this is talking about hands. All right, you could go out like this, right? Where's the thumb? Straight up. It's anterior. Where are the fingers? Going down. They're posterior. So zone of polarizing activity dictates posterior digit formation. Okay, so it's posterior digits, posterior polarity. And so in a developing mouse limb bud, this is a limb bud, you have a zone of polarizing activity. In development, then you get the fingers. Well, in our case, fingers, and the mice, digits. Okay? And you can show that that zone of polarizing activity is important because you can, let's back up here, you can then take this developing limb bud, put it in the opposite direction, and add a zone of polarizing activity. So if you add another one, what would you predict? More posterior digit formation. Is that correct? Right? And when you do that, in fact, that's what you get in the mouse limb bud. So you get more posterior digits in the wrong place. And they know the downstream signals and so forth. You don't need to worry about it. They think it's involved in some pathway involving sonic hedgehogs and the Hox genes and so forth, but you don't need to worry about it for this discussion. The point is, it's a classic positive signal. In the absence of the ZPA, you do not get posterior digit formation. In the presence of the ZPA, you get it. Classic description of a positive inductive signal. Okay? Yes?
So how can you make the thumb?
What?
So
How you make the thumb? There's other kinds of. How you make the thumb? That's an anterior digit. There's other kinds of signals. We're not talking about the thumb. The further you are away from the ZPA, the less posterior digit formation you get. So it's a gradient. So the other part of this is, during development, these signals or hormone signals form gradients. And depending where you are in the gradient dictates sometimes who gets formed when and how. Okay? So that's what kind of gets set up there. Questions on that? Okay. So starting now, since after you develop, the reason I do digestion next is so when you're developing and you're born, one of the first things you want to do is eat. So it's a nice continuation of this discussion. So we're going to start our systems flyby. And some of the concepts, we need to go back to the first lecture for some of this. And drawing, I know I'm jogging your memory here, but if I said to you simple squamous and leakiness, what kind of structure might you think about? Capillaries, exactly. And what happens in the system, it's the ability of this leakiness to how allows all these systems to interrelate. So the brief overview is, here's the digestive system. And as you eat food, you have to absorb it. And where you absorb it is into the circulatory system. Okay? And then these nutrients have to go places, the tissues, and they have an equilibrium. They go into interstitial fluid before going into the tissues, or we'll just say the cells. So this concept of interstitial fluid is absolutely incredibly important for understanding everything that goes on. And these capillaries also dictate how the circulatory system relates to the respiratory system, and how it interacts with the excretory system. And this, obviously, the excretory also interrelates with the interstitial fluid, as you'll see. That's how kidneys function. So the point is, this is all dictated by capillaries. And the important thing is, here's the various capillaries. One capillary. That these molecules can go from inside the capillary to outside. And this leakiness is what gives you the communication in all the systems. So this will be kind of a hidden kind of just concept. Not so much hidden, but you'll see this come across as we talk about the systems. So we're going to talk about digestion first. Why do you eat food? Besides when you're hungry, because you're hungry. But why is your system so designed to make you hungry? You need chemical energy, right? You need carbon backbones, nitrogen, and you need essential nutrients, such as certain vitamins, certain amino acids, et cetera, et cetera, et cetera. That's not something I'll be testing you on. Is that what I just said in the last 15 seconds? So you're eating it for lots of reasons. So before I get into the description of this, I think I'm going to take a slight tangent here and get into sort of the societal, not political, but societal side of this. Is eating and weight control is very important in our society, all driven by hormones, or a lot of it. So just as an aside, but I think an interesting aside, they discovered that adipocytes, or fat cells, actually produce a hormone called leptin. And leptin targets certain neurons, leptin-sensitive neurons. Let's make this bigger here. And as a result, you have increased activity and decrease appetite. Okay, this seemed like really cool stuff because they can take a mouse, they can take an obese mouse, genetically obese mouse, give it leptin. Just give it leptin, and it becomes normal size. And given our society and interest in weight control, when they discovered this, one of the companies laid out hundreds of millions of dollars for the patent to give people leptin as a way of changing weight control. So in other words, the idea is you can eat all you want, you don't gain anything. Not the healthiest thing for other reasons, but clearly something that could play a role in society and for you guys prescribing meds to people. But they then, so this works great in mice, really wonderfully. They started giving it to large trials of people, and it worked for only 1 or 2 individuals out of thousands that were overweight. And so, you know, that money you paid for that patent started going less and less interesting, right? Well, it turns out in humans, we have this kind of system, but we have multiple leptin-like molecules, multiple sensitive neurons, and multiple pathways that get affected. So it looks like weight control in humans is much more complex than mice or rats, okay? So we are different than them. So here's a classic example where you do all these experiments in a mouse, and it doesn't work so well in humans. There's many examples where mice predict exactly what happens in humans, but not in this case. So the point is, it's important to think about, and it's important to think about the process. And you guys will be exposed to this later on. So this is why I like to discuss digestive systems. So what's the generic digestive system? The generic system, it goes through, it's called the alimentary canal. And basically you take food in and get rid of waste. And there's names for all this. But kind of the generic system is as follows. And then we'll go into some details in a second. The generic system is you take in food. This is called ingestion. And it goes into several compartments before you eliminate it. So these compartments are separated by ring-like valves called sphincters. And this is really important. Think of your stomach, right? Your stomach is very acidic. You got to keep the stomach contents away from other parts of the digestive system. So you have sphincters on either side to deal with this. So in this generic system, you can envision bringing in acid and enzymes. These enzymes break down stuff, get pumped into this first compartment where you have food, and that's really called digestion, which is hydrolysis. All hydrolysis is putting water across a covalent bond to break the bond. What you want to do is simply take bigger molecules, bring them down into smaller molecules so you can take them into your body and use them. That's really all that's going on. And that's hydrolysis. So some of this hydrolysis works better at low pH. You then go into the second compartment and you restore the buffering capacity and give it other kinds of enzymes for other kinds of hydrolysis. But in this other compartment, you also want to take the small molecules back into your body, and that's called absorption. And then you have, which is what you want to do, and obviously there it's important that it comes in a very directed way, and that's where, remember we talked about tight junctions and adherence junctions in your intestinal cells. You want to direct the nutrients through the intestinal cells, and then you eliminate everything, and it's called elimination. Okay? If you understand this, you understand the details that we're going to go through here in a second. What? ENZ is enzymes. We'll get to the details in a second. All right? So basically, you break things down, and you take it in. And it's really important to do that. So let's start down here with the human part of this. And we're going to use this metal thing right here as one sphincter. And the other one, this is the other sphincter. So this will be the stomach, and this will be the intestines, and further down. And this will be above the stomach. Okay? And we'll try to put in, oh yeah, one other point I want to make on the generic ingestion. You have various glands and organs pump in the enzymes and the buffering capacity. It's a very important part of this. And you'll see what these are in about 5 minutes. In fact, maybe sooner. Because when you take in the food, it goes into your mouth. The food comes in. And it already sees one of these organs that pumps in glands, and that's the salivary gland. The salivary gland, one of the enzymes, ENZ enzymes, is amylase. Now, what you'll see with these digestive enzymes, you'll see a lot of enzymes, not all of them, but a lot of them have the ASE at the end. This means breakdown. It means hydrolysis. Okay? And you'll see several examples of that. You also see examples where they don't end in ASE. But if you see that, it breaks it down. What if I said to you GTPase? What does that mean? Breaking down of GTP to GDP. There you go. Same derivation of the word. Amylase breaks down complex sugars into simple sugars, or oligosaccharides into simple sugars, such as disaccharides. And I won't finish the name here, disaccharides. Okay? In addition, the salivary gland also has something called mucin. This is from the salivary gland. And mucin lubricates the system and causes helps with the buffering. Okay, so this is also coming in. So both mucin and amylase are being pumped in by the salivary gland. Okay? And what comes out of this, what you start forming, let me just bring this down here. You're still above the stomach. You start forming something called the food bolus. And this food bolus then gets pushed into the pharynx, which we're not going to worry about what it does, the esophagus, which we're not going to worry about it, and takes this food bolus heading to the stomach. Okay? So here we are in the stomach, and I'm going to get a piece of chalk here. You go into this food bolus that goes into the stomach, and you'll see the food bolus actually stimulates some hormonal production. Because the food bolus, okay, this is the sphincter. Here we are in the stomach. And this food bolus comes in. And you have to break this down. So what happens is, in the stomach, you get pumped in from the walls of the stomach hydrochloric acid and pepsin, which helps in the hydrolysis of the food. And you'll get to how this works in a second. And what comes out of the stomach is something called acid chyme. Goes into the next sphincter to the intestines. Let me just bring this up here. Bring this down here. Bring this up here. Okay? So you eat the food. The esophagus pushes the food bolus into the stomach, and you start having hydrolysis. Now, one of the things that takes place here is something very, very important, and that is the concept of making precursor proteins. So what happens is you don't actually pump out pepsin, which is a protease. What you pump out is pepsinogen. And this comes from specialized cells called chief cells that are in the lining of the stomach. This is an inactive protease. Again, the ASE. What do proteases do in general? Break down protein. There we go. It's also larger. What happens is the acid, hydrochloric acid, which gets produced from parietal cells, also in the lining of the stomach, changes the conformation in a way that makes it a little bit active, and you make pepsin, which is the active protease. And it is smaller. So what happens is pepsin starts breaking down all these proteins that are in the stomach. By the way, what do you think the pH optimum is of this enzyme? You guys have pH optimums in other classes, right? What do you think the pH optimum of this is? Yes? Huh? Yes, acidic. I was looking for a more general answer. This enzyme is quite happy in an acidic environment, so therefore it works in the stomach. All the other enzymes that we talk about on this side of the sphincter won't work in the stomach because they don't have the right pH optimums. Okay? So it's a classic example of this molecule that's larger, gets broken off. You get from the inactive to the active molecule. This is active, this is inactive. This is pepsinogen, this is pepsin. You'll see several examples where the ogin, pepsinogen, the ogin, generally that means precursor that's not active or minimally active. You get rid of that part of the molecule and you take off the ogin, you get pepsin, which is now active. It's easy to remember. Larger pepsinogen, get rid of the ogin, now it's pepsin. Okay? And you see there's many examples of this. Larger precursors that are inactive to active smaller molecules. Okay? Any questions? Stomach, you get the food bolus, hydrochloric acid and pepsin start breaking up, undergo hydrolysis, putting water across a covalent bond, breaking the covalent bond. And what you get out of this is the acid chyme. Questions? Okay, so now we go into the small intestines. And I'll put this up here in a second as we continue this. The acid chyme comes into the duodenum, which is the small intestines. And you have a lot of accessory glands, a lot of things go on here. In the duodenum, you have one of the things that goes on is bile gets produced. Bile helps solubilize the fats. And bile comes from the gallbladder, where it is stored there. And that comes from the liver, where you synthesize bile. Okay? So that's getting pumped into the duodenum. Now, if you have a gallbladder removed, what kind of diet do you think you have to go on, generally? A low-fat diet. There you go. Some of you will be prescribing or telling patients exactly that. What also gets pumped in from the pancreas, which we'll go into a little bit of detail here, are a bunch of hydrolytic enzymes. And I call these the X-aces. This is my term. You won't really see that in textbooks. Okay? So what are the X-aces that come rolling into the duodenum? The X-aces are 4 classes of hydrolytic enzymes. One of them are proteases, and there's several. One of the ones that's a very important one is trypsinogen. Now, is trypsinogen a precursor or an active molecule? Trypsinogen, precursor, right. And it gets converted through a proteolytic cleavage. Don't want to go into the pathway to trypsin, which is an active protease. This is one of the most promiscuous proteases out there. It rarely finds a protein it doesn't want to hydrolyze. Okay? And it's made in very large quantities. Comes in from the pancreas. Not the beta and alpha islet cells. This from the pancreatic tissue itself. What do the alpha and beta islet cells make? Insulin, glucagon, right. Those are endocrine glands. What also comes out of these XACEs are lipases, right? Converts fats into fatty acids plus glycerol. Okay? You also have nucleases. What do nucleases do? Yes. Yeah. Breaks down RNA and DNA into phosphate groups, sugars, and bases, right? And the last one is also, there's another form of amylase that continues the process of breaking down sugars. Oligosaccharides, or in this case, the oligosaccharides that the mucin, that the salivary gland doesn't deal with, ends up down here, and these get converted into simple sugars, or di and monosaccharides. Okay? So the reason I call these the XACEs is you got proteases, lipases, nucleases, and amylase. Breaking down of the major types of molecules that are there. Everybody see that? Just an easy way to remember this. But these come from the pancreas. And we'll talk about the hormonal control here in a second. And then you get into the lower parts of the small intestine called the jejunum and the ileum, which are all part of the small intestine. And here basically you have massive amounts of absorption of the nutrients. You also have some absorption in the duodenum, but it's mostly happening down here. Then you have the large intestines. This is what I see, absorption of nutrients and water, where the large intestine is mostly absorption of water. And then you have elimination. Okay? And I'll put this up here so we have it in order. And put the... All right, so how many of you guys have had breakfast today? What hormone is coming out? Insulin, right? Changing glucose. But in order for glucose for your body to function, you've got to have a regulation of this. So you guys have eaten your food. You've pushed the food bolus into the stomach through a sphincter. You start breaking it down in the stomach. You make this acid chyme. The acid chyme goes into the intestine, induces the bile formation. The XACEs, proteases, lipases, nucleases, amylase, breaks everybody down. Hydrolysis, small molecules, water killing, water going across covalent bonds. And then you absorb like crazy. You have to have this system coordinated. You can't just eat and say, oh yeah, am I going to start digesting or not? You want this whole thing coordinated. So the hormones can do it. We'll do that for you. So, in the stomach, when the food bolus comes into the stomach, hits the lining of the stomach wall, you form a hormone, gets produced called gastrin. And gastrin binds to its receptor in neighboring cells and causes these neighboring cells to produce the gastric juices. Is a generic term, but what it is hydrochloric acid, liquid, enzymes, et cetera, et cetera. It just starts pumping stuff into the stomach so that you can start the hydrolysis process. It gets kicked on by the food itself, food bolus, hitting the stomach wall. What kind of hormonal regulation is this? Paracrine, autocrine, endocrine? Paracrine, very good, because it's not going through the blood, it's going from one stomach interlining cell to another one. So it's a classic paracrine system. Does everybody see that? Okay, so we're bringing back concepts we learned. In the small intestines, the acid chyme coming in also kicks on some important hormones that help with this whole process. So the acid chyme comes in to the duodenum and causes the production, one of them is a hormone called secretin. Oh yeah, one other point. I missed one point here, which is very, very important. That the pancreas also produces bicarbonate. Let me just put this up. I'm going to make this very clear here. Pancreas also produces bicarbonate. Why do you think it makes bicarbonate? Bicarbonate is a base. It buffers the acid from the stomach. Exactly right. This is very important in this process. Okay? And I'll put this back up here. So it's a pi. Secretin gets produced from the intestinal walls, duodenum walls. Secretin binds to its receptor. Remember, everything is receptor driven in the pancreas, and causes the pancreas to secrete bicarbonate. So it's on cue. The acid chyme causes the production of the bicarbonate. Very important to see that. In addition, the acid chyme comes in to the duodenum wall, and you produce another hormone called cholecystokinin. What cholecystokinin does, it binds to a receptor in the pancreas, and it causes the pancreas to release the X-aces. Okay? As a generic, it kicks out all these enzymes. In addition, cholecystokinin goes to the receptor in the gallbladder and causes the release of bile. So this system is completely coordinated. You eat the food, you get this food bolus, you cause the stomach to release gastric juices and enzymes, hydrolytic enzymes. You go into the small intestine, the acid chyme causes production of a hormone, secretin, to neutralize it, which is critical, because if you didn't, your intestines would get eaten away. In addition, the acid chyme comes in, produces another hormone called cholecystokinin that causes the pancreas to release bicarbonate and the X-aces, the hydrolytic enzymes. So you produce these hormones and you produce the X-aces in the hydrolytic capacity when you need it. How many of you haven't eaten yet today? Right. That's not happening with you guys. You don't want the X-aces or the gastric juices really rolling until you have food. So when you eat, you tell your body says, here's insulin, let's bring in the glucose, let's regulate it. But in addition, let's hydrolyze everything so you can actually have energy, carbon backbones, and survive. So it's all completely coordinated. Okay? Any questions? So, yes?
Which pathway does the secretin take to go to pancreas?
Oh, very good question. These are primarily paracrine type pathways. There might be a little bit of blood involved in some of these. I don't know the exact connections between them, but you can view it as paracrine.
Okay, so paracrine can happen when they're on different organs as well.
Yeah, oh yeah, if they're next to each other. There might be a little bit of capillary action. I'll check into that. Not critical to understand. All right, so this weekend when you guys study, and I remember I would, in college, I'd have 5 meals sometimes, as I stay up all night. and wait till the last moment. Just remember, when you have that 5th meal at 5 in the morning, all this is going on as you study. Good luck with your studying.